Hi, welcome to the fourth installment in my fourth lecture on the coronavirus. This lecture is devoted to the immune system and how our bodies respond to a coronavirus infection. In the first installment, I covered the differences between the innate and the adaptive immune system. In the second installment, I discussed how the innate immune system mounts a generalized inflammatory response to combat infection. In the third installment, I described how the innate immune system mounts an intracellular response to block the replication and propagation of viruses from infected cells to prevent an infection from spreading. And in this installment, I'll discuss the adaptive immune system. Now, the adaptive immune system is one of the most amazing things in the universe, second only in complexity to the human brain. It has an amazing, challenging, difficult task to execute on, and that is it has to be able to recognize any potential pathogen that might exist anywhere in the world. And that includes not just now, but any pathogen that might exist in the future. It has to be able to detect those pathogens and mount a response to help eliminate those pathogens. And it has to do that without damaging any of our own cells. So it has this amazing, incredible challenge of having enormous breadth, while at the same time, so it could detect anything, while at the same time having tremendous specificity so that it only attacks foreign things. And so I'm going to get into, in this lecture, I'll start getting into how that works. And first I'll kick this off by talking about the concept of shape complementarity. This is very important to the function of the adaptive immune system. So what's the role of shape in specific binding? And we encounter this concept in our everyday lives all the time. So just like proteins can interact, macroscale physical objects can interact. So here I have a baseball, I have a candlestick holder. These can interact, but you can see they don't have good shape complementarity, so the interaction is unstable. In biology, we would call this a weak interaction or a low affinity interaction. However, here's the thing that really normally interacts with the candlestick holder, a candle. The diameter of the candle matches the diameter of the hole in the candlestick holder. So this interaction is a much more stable, tight, strong. We call this a high affinity interaction. All right? Now, just to give you another example, here I have a couple jars. I have a lid. This lid can sit on this jar here, but it's not a tight interaction. It's a weak, unstable, low affinity interaction. But if you widen the mouth of this jar a little bit, as is found on this jar here, now the lid forms a tight, stable, high affinity interaction where it doesn't easily fall off. And as we'll see, that's going to be really important, that idea, in the adaptive immune system. The other thing I'll introduce you to are the key cells of the adaptive immune system, and that would be the T cells and B cells. Now, these are collectively called lymphocytes, and altogether in the human body, there's about roughly 2 trillion lymphocytes, so that's 2,000 billion. That's a huge number of cells. If they were all in one place, they'd be about as big as the brain or the liver, but they're not. They're spread in different parts of the body, in lymphoid tissues like bone marrow, like spleen, like lymph nodes, so that they can better surveil extracellular fluids to look for signatures of a viral or a microbial infection that they could then mount a response to. Now, T cells come in two broad flavors, the so-called helper cells and the cytotoxics. And I'm going to focus on the helper cells in these lectures. These, they don't directly engage in the event, in the battle, if you will. They're more like coaches, where they interact with the other cell types that are engaging directly with the invaders, and they instruct them, they help them mount a more effective attack. The cytotoxics, on the other hand, they directly go up to cells that are infected with microbes, bacteria, or viruses, and they kill them to prevent that infection from spreading further. But as I will focus on these T helper cells. Now, a key thing for T cells and B cells is that they have unique cell surface receptors that define them. And for T cells, those are called T cell receptors. And for B cells, they're called B cell receptors. And every T cell and every B cell has a unique T or B cell receptor. So if I take two different T cells, they're different because each one has a different T cell receptor that recognizes a different thing. The things that these receptors recognize, we call those antigens. Now, I have to introduce you to explain how T cell receptors work. I'm going to have to introduce you to two additional concepts, or two additional things. The first is dendritic cells, and the second is MHC, major histocompatibility complex molecules, which are proteins. Now, dendritic cells, they're essentially like a vacuum cleaner. They go around in tissues and they suck up dirt and detritus and debris in the environment. They bring it into the cell and they break it down into little pieces, and then they put those pieces on the cell surface for display so that the adaptive immune system could get a sense of what the dendritic cell encountered. So I like to think of dendritic cells, they're like the Roomba, right? They're on autopilot going around vacuuming stuff up. Now, here's a dendritic cell that I've shown here. And this dendritic cell is constantly taking up proteins from its environment, bringing them into the endosome. So here I've got three proteins, the square, the circle, and the triangle. It brings them in the endosome, and then once in the endosome, it breaks them down into tiny pieces. So let's imagine the pieces are the same shape as the protein they came from. So tiny triangles, circles, and squares. Now these tiny pieces get loaded on MHC molecules, specifically what are known as MHC class II molecules, in the endosomal membrane. So we see here the mini triangle, mini circle, mini square. These then get displayed, they get sorted to the cell surface and displayed on the cell surface. So, you know, we imagine we have our molecule here with the square, we have one here with the circle, and over here we have one that's carrying the triangle. Now, every T cell has a unique T cell receptor, and this T cell, this T helper cell, happens to have a T cell receptor that recognizes an MHC molecule that's loaded with a mini triangle because it has the proper surface complementarity. Now, imagine that these other two molecules here, the circle and the square, that these molecules come from normal cellular proteins. So in that case, there will not be a T cell receptor that recognizes them, because any T cell receptor that recognizes one of our own proteins is eliminated during development. And that's a safety valve to prevent T cells from arising that can attack our own tissues. So they can only recognize foreign antigens, and we'll imagine this triangle comes from a viral protein. Now, another thing I need to state is that the dendritic cell is constantly sucking things up and displaying proteins on its surface, but it's really only going to go to T cells when it has a second stimulus that would indicate there's a pathogen. So for example, it detects a virus, just like we discussed in prior lecture for innate immunity. That virus turns on the dendritic cell, and then the dendritic cell says, oh, you know, one of the proteins I sucked up and presented on the surface may be a viral protein, so I should go warn the adaptive immune system. So it leaves the tissue. In this case, it might be the lung, in the case of coronavirus infection. It enters into the lymphatic circulation, and it goes to the nearest lymph node. And the lymph node is jam-packed with T and B cells, and it encounters T helper cells there and displays the antigens it found. And if it found an antigen that comes from a virus, and if there's a T cell receptor specific for that antigen, you now start an adaptive immune response. And I'll be back with the next segment in which I discuss B cells and B cell receptors.